Thank you, Erika, and thank you to all of you for being here today. My name is Teemu Miettinen, and I work in the laboratory of Scott Manolis right here in the Koch Institute. In our lab, we study cell division from a physical perspective. We want to understand how cells regulate their mass and their stiffness, and in this case, the movement of their plasma membrane. What you can see here in this image are two cervical cancer cells that have just divided from each other, but they are still connected by a narrow bridge. The plasma membrane, which is the outermost layer of a cell, is a highly fluid structure that moves around the cell like the arms of a dancer as the cell divides. And here in this image, you can see the plasma membrane in red and yellow. In blue, you can see the DNA of a cell. So that would be all the contents that tell the cell what it is and what to do. In a normal cell division, the DNA is segregated evenly between the two daughter cells. However, sometimes cell divisions fail and the DNA doesn't segregate evenly. This means that mutations arise. Such mutations can cause cancer or sometimes just cells die. But to help you understand what this really looks like, let's zoom in to the cell division on the right-hand side. Here you can see two cells that have just separated from each other, but a narrow strand of DNA can still be seen running between the cell in a very thin bridge that connects the cells. Such lacking strands of DNA will eventually have to be cut in order for the cell division to complete. Now, consequently, one of the daughter cells is going to get excess DNA that the other one is missing. Both of the daughter cells become mutants. Both can behave abnormally. Now, in a normal human body, there will be more than a quadrillion cell divisions in a lifetime. Even more if you're unlucky with cancer. Now, that number is huge. It's more than stars in our galaxy. Okay, it's way more than I can really imagine. It's 10 million billion cell divisions that your body will undergo. Now, most of your body's biomass it's in your muscles, it's in our adipose tissue, but most of cell divisions are not actually for those cells. Most of the cell divisions in our body are taking place to form the very small and short-lived cells of our blood, so the white and red blood cells. These cells, due to their small size and their short lifespan typically, are made in billions every single day. And if we look at cell divisions in cancer cells that originate from your blood cells, such as leukemias, we see cell divisions that look very different. So these leukemia cells are pulling their plasma membrane to the side of cell division as their two daughter cells separate, so that by the end of cell division, the space between the two cells has this excess accumulation of plasma membrane that we do not see in most other cell types. And if we image these cells with even more resolution using an electron microscope, we can even see how the plasma membrane is folding around the bridge that connects the two daughter cells. Different cell types, vastly different looking cell divisions. Now, we believe that understanding these physical differences between cell types will one day enable us to actually prevent cell divisions in a specific cell type, such as these leukemia cells. But to do that, we have to first understand how cell divisions look like, and we have to understand how different cell types achieve different looking cell divisions. In our case specifically, this means that we want to understand the molecular mechanisms that are pulling the plasma membrane to the division site in the leukemia cells. And once you understand those mechanisms, you can start to perturb them, not only to understand cancer, but potentially to treat cancer in diseases like leukemias. This is how basic cell biology lays the foundations for cancer research. And this is why even here in a cancer research institute, we have to understand cell biology. So with that, I just want to acknowledge that this work would not be possible without great supervision by Professor Scott Manolis, our wonderful collaborators, and very importantly also our generous funders who understand that we have to sometimes look at the very basics of biology before we can do cancer research. Thank you for your time.